All right, thanks for coming on this pretty cold, blustery day. So I'm going to talk to you a little bit about how we track animals through space and time, and then really try to use that information of tracking those animals to understand how populations respond to changing landscapes. And this is really, there's not a lot of final results here. This is really more of an idea of how we think about how animals interact with landscapes, how we study them, and how we can ultimately make management decisions to basically promote the continuity of these species in our landscapes. So all animals interact with landscapes at different spatial and temporal scales, and it can range from extremely massive in the case of that albatross in the top right to very small for the mussels down the far left. You know, that albatross might go over many degrees of latitude and longitude within a single year, whereas that mussel will spend most of its adult life in a few square centimeters. But of course, as a pelagic larvae, it goes an unknown distance in the landscape or in the seascape. Now, how animals interact with these landscapes, it really matters. You know, we can think about this classic image from Senft in a 1987 bioscience paper that I think really illustrates well how at least herbivores sort of interact hierarchically with landscapes. You know, there's this very broad-scale choice of where a species might exist, sort of the habitat suitability for a species, or essentially the niche of that species in space. And then within that area that's suitable, animals are going to choose areas of home ranges where they're going to spend their life to extract resources, find mates, and basically spend their lives. Of course, within that home range, they move around. They choose places to forage, and at places that they're foraging, they actually make decisions of what to eat, you know, choosing amongst a different plant species at that actual bite level. And you know, most of the time what we're studying is, you know, we look at how these animals move around, where they are in the landscape. What is their home range and where they move around in that. But at the end of the day, I think what we're really interested in is what's going on here. And it's something that we really can't measure very well. I mean, we all know that you are what you eat, right? I mean, so what the animal's actually chewing on makes a difference to its health and longevity. But how do you get some inference for what's going on down here based on what the animals are doing at this broader spatial scale? Because it's down here, it's where the rubber meets the road in terms of where the animal, an herbivore at least, is extracting resources and how it's going to ultimately put on fat for the winter if it lives in northern hemisphere, for example. So as an example, this is Equus hemionus, the kulan in Mongolia, the Gobi Desert. This equid is existing in what I can only describe as what must be a terrible place to be a browser or grazer, rather. I mean, it just does not look good, right? I mean, and you see the consequences of this in the animals' movement patterns. I mean, these animals move vast distances day to day. You know, they have to basically exploit this very low-quality resource or landscape for resources of food and water, because it's also very dry. And so how they do this, how they interact with that landscape spatially and temporally, behaviorally, makes a difference to whether that animal will live or die, whether it will or will not reproduce. And this matters for this species. Understanding how that animal interacts with the landscapes matters because those landscapes are changing. You know, in Mongolia, you're having increasing conflicts between the wild herbivores and nomadic grazers. And so you have those human-wildlife conflicts. People don't necessarily want these wild species out there. There are fences being built for a variety of reasons, train tracks being put in. All this causes conflict for a species that is, you know, basically declining now. Okay? And so understanding how that animal interacts with the landscapes can matter in terms of how the government decides to allow fences in one place versus maybe not in another, and thus promoting the ability of that animal to successfully navigate this landscape. Now, this is a very different herbivore and a very different landscape. I mean, this elk has its muzzle down in a bunch of green. I mean, the world is green to this elk in Yellowstone. But as this animal is grazing, it's making a lot of choices. It's choosing amongst different plants at that bite level. And ultimately, it too will have to go through a very harsh season, in this case, wintertime in Yellowstone, where it doesn't have access to good forage. And so, again, the choices it makes, the patches it chooses to extract resources from will really matter. And then finally, the species I'll talk about most today, the beleaguered Minnesota moose. This is a moose, trying to make a living in the frozen landscape of northern Minnesota. There's not a lot here for these animals in the wintertime. They're mostly, browsing twigs from shrubs and trees. And again, understanding, where does this animal move? How does the animal use space? How does the animal change its diet based on what's available to it so that it can get through till spring, until green-up comes? And then more to the point, in the summertime, how does it efficiently use that landscape so that it gets enough food to get through that next winter? Because these landscapes are changing, okay? We all know that land use and land cover are changing across the globe. This is just an example in Minnesota, some historical data of housing density and some projected data for 2030. And, fragmentation is increasing in the habitat. Patterns of logging and other resource extraction are changing based on economic drivers. And so understanding how animals are responding to this change in landscape pattern can be really important to understand if they can actually make it through in the future. And this is especially important because this land cover change, which is happening quite quickly, is taking place in the context of a slowly changing climate. And especially when you're a moose and you're built for cold, warming temperatures are not good for business. Okay, these animals, they suffer even in the wintertime sometimes here when they have their winter coats on. They have to try to spend extra energy to expel heat, even in January sometimes in Minnesota, because it's not quite cold enough for them. And in fact, here they're basically at the limit of their range in terms of the temperatures that they can withstand. The maximum temperatures in the summer and the maximum temperatures in the winter are really quite close to the limit of what we believe that species tolerates. And so as temperatures warm, how does this animal change its behavior to try to mitigate the effects of that thermal stress? And does that change in behavior yield a change in diet that may be linked to the very well-known population decline of the species in the state? So if you actually start following animals around, regardless of the animal you're looking at really, you see that these animals aren't moving randomly. They're basically moving around and returning to spots, going to other spots that they like, spending some time there, and then going elsewhere, coming back. And if you actually watch this, try to switch back there. If you actually watch, this is an animation of an actual animal. It's an elk moving through a landscape. But you can see that the animal doesn't move randomly. And you wouldn't really expect it to either, right? So the animal will spend some time here, and then it zips across over here. And as you see it moving around, it's basically going back and revisiting places it's been before. And these animals are 8 or 9 years old, perhaps. They know this landscape very well, so they know how to extract resources efficiently. And so understanding this resource pattern is really, really important. Understanding how the spatial structure of that landscape basically interacts with the movement behavior will help us understand and make predictions about where that animal will go in the future. But I would argue that that's not enough. We don't want to know just where those animals are going to go in the landscape. Resource managers and conservation ecologists, we really want to make predictions about how animal populations are going to respond to these novel landscapes and climates, these novel combinations of land cover, combinations of land cover with climate. So to do that, we asked several research questions. First, how do populations interact with disturbance regimes and respond to broad-scale environmental changes? How are the movement and distribution of animals influenced by predation risk and heterogeneous resource distribution? And then finally, this is the real key one here. How do these movement behaviors of animals interact with landscape pattern to feed back and affect population dynamics? How does where an animal goes affect what it eats, which affects its mortality risk? Understanding that, and understanding how we can manipulate landscapes if necessary to change those demographic parameters, I think is a really key goal for a lot of wildlife ecology. So, going back to the moose here, the moose population's been declining substantially. I mean, since 2005, it's declined by an estimated, I think it's around 60%. It's not doing well. The moose population in northwestern Minnesota declined from quite a large population down to less than 100 about 10 years ago. And it's they're still out there, but not very many. And our northeastern population is the one we're really worried about now. And of course, why these animals are declining, there's lots of reasons. It's complicated, right? I mean, we talked about this thermal stress in the summertime and possibly the winter, but there's also that winter energy limitation. They don't have high quality forage through that winter, so they're burning fat and muscle, trying to make a living off of those twigs, which is not really great food. And meanwhile, they're sort of interacting or responding to or being taken advantage of by predators and also parasites. Wolves are going to eat the moose if they're available. Parasites are going to infect the moose if they can. And I would argue that all of these features are influenced by the spatial distribution and abundance of high-quality food and cover. So if you have a really high-quality food landscape that's adjacent to a good cover landscape, that's probably a good place for a moose. And so it could be that the distribution of those cover types, if it's very dispersed as opposed to very clumped, might make a difference to how those animals make a living in those landscapes. So if we look at the broad-scale population trend here, this is essentially the main moose range in Minnesota right here. It's the area that actually gets surveyed annually by the DNR. In aerial surveys, they don't survey this whole area, but they subsample it. But in this area, this is the population trend with some confidence intervals. Again, you can see that decline is not looking good for the moose. It has been noted that correlated with this decline has been a decline in board feet extracted from the landscape by the forest industry due to economic reasons. And so, one of the ideas is, well, maybe there's just not a good enough habitat in this region to support that population, and that's one reason it goes down. But I'd argue it's not quite that simple. If you were to break this up into different regions, in this case 3, it's possible that all 3 regions the population is going down. I mean, this is just a cartoon to demonstrate that. But an alternative is that a few of them are going down, and maybe one area is stable or going up. Okay, we don't know right now. We haven't actually built those spatially explicit population models yet. And you can go even further. If you go down to the level of the actual moose sample unit, which is what you see here, these are basically 2 by 5 mile areas that are sampled, again, subsampled by the DNR every year. There could be many populations that are declining and a few that are doing all right. And if we knew that there was some kind of landscape feature that was linked to those areas of decent population, that might give us some real grist for the mill when it comes to how we actually make management recommendations at a broad spatial scale. So the question, though, is how can we really build mechanistic links between local population dynamics and the distribution of resources? So how do we make that link of where animals go and how well they do, okay, how well they survive and reproduce in those areas? Well, you have to start with knowing where they go, right? So you do what we do, which is put collars on critters. This is a mask to make sure the animal doesn't stress out a lot while we're putting on those collars. And the DNR deployed about 130 collars in conjunction with the Grand Portage Band of the Lake Superior Chippewa and also the Park Service. So we have about 130 some animals, or had 130 some animals on the air earlier this year. We're down quite a few due to mortalities. But the idea is that you track these animals through time, okay, and across space. and you look at where do these animals go? Where are they spending a lot of time? And also, when and where do they die? Okay, this is an example of an animal that was taken down by wolves. They very politely left the collar for us around the neck of the animal, and this was one of mine, and I'm very glad they didn't chew through it. They're not cheap. And so this is an example where the wolves really did finish their meal, but it's not always this simple. Sometimes an otherwise healthy animal just dies. This is an animal up near Grand Portage that on Labor Day weekend this year just tipped over on her side and died. There was no reason that we can understand about why, what actually killed this animal. This is where she finished off. So again, understanding what did that animal interact with here that led to that mortality, that rather strange mortality at the end of her road. Well, again, one thing you can go to is how much land cover is actually decent quality habitat for them. This is just proportion of forest in each of these moose survey blocks, where dark green indicates high proportion of forest and light green is low. And you can see there's some heterogeneity across this moose region. What we can do, though, to try to understand how that cover changes through time, how the amount of, for example, moose forage might change through time, is we can actually look at the forest inventory and analysis plots in Minnesota. These are plots that were established by the U.S. Forest Service, and they're revisited, I think, every 5 years. And if you start looking at how many of these plots are within those, each of those survey blocks, you can start seeing, looking at those points as subsamples, and this is the percent of forest that's actually mature. So in other words, of the forest that was sampled within a block, what proportion of it was mature forest? And perhaps good thermal cover, but maybe not good food supply. And then you can look at the change. This is the change, the delta in percent forest mature between 2005 and 2012. And gray is indicating basically no change. Yellow is negative, so the forests got basically younger, and green is positive, meaning that more of the forest got older. And I guess what I want you to see from this plot, this is just a preliminary plot. It's not extremely accurate at this point, but it gives you an idea of the heterogeneity in what's going on in our forests. But it's still at a pretty coarse scale. We can also zoom in a bit and start looking at classified land cover images. So this is a Landsat image that was classified in 1990, and then here's 2000. So I'll go back and forth a few times there, and you can see that the cover types are changing. Those brown colors are basically shrub layers. The green is forest, dark green being coniferous, light green being deciduous. So you can see that there is a change, especially in this area here of this particular plot. How much does that matter? The amount of change and also the arrangement. Well, what we have to understand is what kind of food and cover are available. And we have to go out there and actually sample these things, do shrub plots and overstory plots, calculate how many seedlings are out there, possibly how much herbaceous biomass is out there, and then make statistical models that make predictions across space about how much food and cover you might expect to find in these different cover types across the landscape. But this is the real link here. We want to understand what these animals actually eat. And again, I mentioned earlier that this is a little tricky. You can't spend a lot of time, especially in northern Minnesota, with binoculars watching what these moose are eating. The vegetation is thick, you'd never get very far. But what we do is when we capture these animals, we pull a little bit of hair, not this much, this is just an image, but a few of these hairs from the withers region, basically the nape of the neck of these animals, and we can segment it. And by looking at stable isotope compositions of this hair, we can get an idea of what that animal was eating in the springtime versus the fall. So the new coat, ungulates serve at least blow off their coat in the springtime, regrow it over summer, and it stops growing more or less in the fall. And so we have some idea here what's going on, what they're eating in the springtime, and here what we're eating in the fall. Because we've also sampled a variety of forage species, we know what their stable isotope signatures are too, and that shows up in the hair. And so once we know what they're eating, and once we know where they go, we can build these mechanistic models of how those animals are interacting with the landscape and how they're extracting resources. And that gives us some idea of how those landscapes are influencing the condition of those animals. And we can make predictions about where they might go based on the levels of selection. So if an animal is starting here at this triangle and then making some decision to move like the next hour, maybe it's selecting for this white area here. Well, if there's no selection at all for that, it's just going to sort of randomly walk around. Okay? But as that selection increases, you can see that that relocation kernel, which is what you're seeing here, this probability of relocation in different spots, basically increases. It starts to track onto that resource distribution. But importantly, these animals don't move at light speed. They're also avoiding these areas down here. This is good habitat, but it's too far away to be really relevant to the animal. You can take this and start building more complex models that take into account memory and revisitation of sites. And it gives you can build different models to describe where animals go or where they might go in the future. So very briefly, I just want to discuss some of our initial pilot study to try to make that mechanistic link between where animals go and what they eat. So in Minnesota, temperatures have changed. This is the modeled temperature in the region from the 90s. This is the 2000s or aughts. You can see that those temperatures have gone up. And in fact, even if you look now across the region of northeastern Minnesota, known as the Arrowhead, there's about a 5 degrees centigrade mean maximum summer temperature in this area, going from cold up here near Grand Portage, moderate in this area, and then over near Ely is what we call our warm plot. So it's about 5 degrees warmer than the area by Grand Portage because you have a lot of temperature mediation by the lake. Interestingly, this sort of flips in the wintertime. We have established a bunch of vegetation plots, which are shown by those yellow dots. We've sampled hair, actually more than you see here in these stars from animals at capture. To try to get at some of, or try to control for some of the environmental heterogeneity, because that's a huge area that we're trying to study there. There's changes in soil, there's changes in temperature, there's changes in vegetation community. We're doing some experimental control in conjunction with the B4 Warmed project, where they're actually maintaining areas at ambient plus 2 centigrade, and ambient plus 4 centigrade. And in those experimental plots, we're actually measuring how does changing temperature affect the amount of secondary compounds in these plants and also the stable isotope patterns. Because one of the hypotheses is that as these temperatures warm, these secondary compounds that might, for example, bind with protein and make food harder to digest might become more prevalent. These compounds can also make food taste not as good, right? So they may not be as favored by the moose. And so we're actually going to go, we have gone out there and started to look at how temperature is impacting isotopes and stable and secondary compounds. And then, of course, the fun part, which is the field sampling. This is a few of our undergrads that were out in the Boundary Waters last summer with my student John Barini, and they sampled a ton of sites to try to get at spatial variability in these stable isotope compositions. And they worked really hard at that. Next summer, we hope to go out and actually measure the community structure in those same areas. So some of the really interesting results. So this is from the B4 Warm study. And this is the number of secondary compounds in balsam fir. The yellow represents ambient, blue is ambient plus 2 centigrade, and green is ambient plus 4. And what you see in this Venn diagram is the number of shared compounds is really high. So 403 shared compounds in those plants. And we don't know what those compounds are yet. That's the second rate stage of analysis. But what you see here is that there's a pretty, there's 10 to 16 unique compounds across those different temperature regimes. Most of the compounds, though, are common. That's okay, because these animals don't really eat balsam fir on the mainland. It's one of their least preferred species. They'll eat it, but they don't like to. One of the things they do like to eat, though, is paper birch. And the curious thing you see here is, one, there's a lot less secondary compounds overall, but more importantly, in that warmed area, this one of the most preferred species for moose has a lot more secondary compounds unique to it, meaning that those plants are spending a lot more energy to put in defensive compounds. This can have profound impacts upon how that plant is digested by moose, and it can ultimately impact how much energy is extracted. So this is a fairly complex diagram showing the isotope results from plants. This is all winter forage. Each of the colors represents a different species. Just for example, this light blue is paper birch, and let's see, this orange here is, or I should say the red here is aspen. And so what you see here is the delta 15N concentrations on the y-axis and carbon, delta 13 carbon on the x-axis. And all I really want you to see here is that these plants have different signatures. Okay, so how we can take some plant matter and start plotting it on here and know what species it came from. And likewise, we can take the animal's hair and start looking at what those animals actually eat. And if you look at the results from some of our moose hair analysis, we basically clipped it at the base, so the most recent grown hair, and at the tip, okay, the oldest hair that was grown in the spring. And if you just look at the change in, or the nitrogen concentration and also the delta 13 carbon, you see that at the base here, which is fall, there's not much difference between what's going on in the spring at the very tip. But if you look over at carbon, there's a lot more variability amongst those animals here in the fall compared to what's going on in the spring. And this is consistent with what you'd expect to happen as these plants got older through the seasons, or through the season. They're starting to put on more defensive compounds in the end of the year, and how much variability there is across space. And there's a strong spatial component to this that we tested for. That can really impact how much late season forage these animals are able to take in. So going back to that initial sort of hypothesis slide, there's a lot of stuff that's impacting this animal. And these landscapes are changing either through forest fires or through logging, through insect defoliation. And at the end of the day, populations really are made up of individual animals. Okay? And how an individual interacts with a landscape is the signature for its behavior as an individual, but also possibly of the species. And if we can have some idea, but consistent patterns of how those animals interact with very dispersed resources versus very clumped resources, we can start to have some idea of making links to the broad-scale population dynamics. Do animals that live in landscapes that look like this have a higher probability of mortality than animals that live in areas like this? And given that we've had a rate of about 20% mortality of our collared animals, this gets us somewhere. We can start looking at this through time. So the next steps of this are really to try to understand how animals alter their behavior in changing landscapes. Ideally in an experimental context where you can actually alter forests before and after and look at animals' movement patterns and forage behavior before and after that manipulation. And then link these movement patterns to diet and condition using the stable isotope analysis, and also by tracking these animals in the wintertime and actually collecting their urine in the snow and isolating their body condition based on that. And then finally, this is the ultimate goal of all this, is to make broad-scale predictions about how what's available to the animals in the landscape, not just in amount, but also in composition and arrangement, how does that interact with their movement patterns to affect the survival of those animals and also their fecundity? So I'd like to briefly acknowledge my collaborators here, folks in the Forest Resources Department. It's also Earth Sciences up at NRRI. The Minnesota Department of Natural Resources has been a huge help. I mean, this is basically their project that I'm working on them with. And also folks in the Grand Portage and Fond du Lac bands, the Lake Superior Chippewa, 1854 Treaty, and National Park Service. So thank you very much for your attention. I'll take questions.